Hello, I am doing my written section presentation. And for question 1, it's got to indicate whether the lac operon will be inducible, non-inducible, or constitutive. And for the partial diploid strains, we're going to describe the overall phenotype of the cell. For part A, our chromosome is going to be constitutive because the lac repressor will not be being made, so the gene will always be turned on. For part B, it'll be non-inducible because the lac repressor can bind to the LAC-O site, but it can never be released because the allolactose will not bind to the repressor to release it. For part C, it's going to be non-inducible because the promoter site, which starts the expression of the lac operon, is not working. For part D, it is going to be constitutive, and I don't need to read my reasoning why. It'll just help you guys grade it faster. And that's kind of the reasoning and also the phenotype or what's going on in those things. For part E, it's going to be inducible. For number 2, we want to use DNA sequencing, the Sanger method, to determine the nucleotide sequence of the DNA molecule shown below. And so the one that's going to be made in the PCR is going to be the complementary strand. So I haven't read the complementary strand to the one that we want to duplicate. And so they want to know what the banding pattern will look like. And this is what it'll look like right here. We start with the smallest segment of DNA going further, and that's going to be G, and then C, A, T, A, A, and it's just going to be going in this order from G, from the 5' end to 3' end, so G, C, A, T, A, A, C, and then T will be the longest at the back right here. All right. And for number 3, they give us the mRNA codons, and they want us to find the all possible tRNA anticodons. And so we have the mRNA codon 5' end GCC and a 3' end. So we go down here to our little wobble rules chart, and we look at the mRNA codon for C. So we see that it can have a G or a, U, and I. So we have G, A, U, I for the 5' end and the tRNA anticodon. For part 3B, and that 3' spot, have a G. So we look at the G's, we know that the anticodon can be C, A, or U. So we put C, A, or U. C and D, it's the same thing except they give us the tRNA anticodon. They want us to find the mRNA codons. So this one, we're going to be looking on the 5' side. It's going to be the wobble end for the anticodon. And so U, if you go over to the chart, can be either A, U, G, or C. So we have A, G, U, or C for the mRNA codons. And then for D, it's A. So we have U, C, G, or A, U, C, G, or A. And that's how you find out those. They're pretty simple. They can be a little tricky, though. For 4D, they want to know the length and nucleotides of the portion of E. coli mRNA that translate your favorite gene. And we figured out that from the paper, that our gene weighs 75 kilodaltons, and that each amino acid is about 125. So we have 75 kilodaltons divided by 125 daltons will give us one amino acid, and there are 3 nucleotides in each amino acid, which would give us 1,800 nucleotides. For 4b, I actually got this one wrong on the test, but later looked it up, and the 5' UTR and is the untranslated region, and that codes for proteins which help modify and regulate the transcription process, and then the 3' UTR codes for proteins which help modify the protein that gets translated from the coded region. So those transcription. For 4c, they want to know how many ATP molecules are required to translate 100 copies of your favorite gene, your favorite protein. And so per nucleotide, we use 1 ATP, and there is 118 nucleotides per copy. And so we times that times 100, and we get 180,000 ATP molecules. For part D, they want to know how many DTP molecules are used to translate 50 copies of your favorite protein. And so it's 2 GTP per nucleotide, and we have 1,800 nucleotides and 50 copies. And so to translate all those, it's going to cost the same amount as before, but you're actually going to, since you're making 50 copies, it takes 1 GTP to end translation, so it'd actually be 180,000 and then 50 GTP molecules. And for part C, you would have 100 extra ATP molecules that I have as the answer for this, because it takes 1 ATP to end translation. All right, and that's all. Thank you for the semester. Have a good summer.